to my channel BKN World of Insects. I am Bhumika. In today's video we will be talking about order Lepidoptera and its basic identification features. Lepidoptera have over millions of years evolved a wide range of wing patterns and coloration. Accordingly, this is the most recognized and popular of the insect orders, with many people involved in the observing, studying, collecting, rearing and commerce of these insects. It is one of the highly specialized insect distributed worldwide. It includes scale-winged insects of the Holometabolous Endopterygot series. Many species of the order are of economic interest by virtue of the silk they produce, or being pests, or due to the pollination they carry out. So they play a very significant role in ecological cycle. Many lepidopterans are valuable in biological research, including work in ecology, biogeography, systematic, genetics and physiology. Recent estimate shows that this order has more than 170,000 known lepidopteran species from 124 families across the globe. Also, recent estimate of the diversity within Lepidoptera from India reveals that the group comprises over 1500 species distributed over 84 families and 18 superfamilies. From the work of many taxonomists from Gujarat, it is estimated that over 193 Lepidopteran species are there in the state. And when I was doing my study of PhD, I was able to find and identify nearly 88 to 90 species from the city of Baroda and its surrounding in 2008. This is the recent classification, which mentions 4 suborders: suborder Zeugloptera, Aglosseta, Heterobathmina and Glosseta. These 3 suborders, Zeugloptera, Aglosseta, Heterobathmina, each comprises of one superfamily and one family. And suborder Glosseta, it has 6 infraorders: Decnonypha, Acanthoctesia, Lophocoronina, Neopsiostina, Exoporia and Heteroneura. Decnonypha, Acanthoctesia and Lophocoronina comprises with one superfamily and one family each, and Neopsiostina, it has one superfamily and 2 families. Exoporia has 2 superfamilies and 6 families. Heteroneura has 8 clades, 37 superfamilies and 124 families. Out of that, Dithyria and Monotricia are 2 of 8 clades. The suborder Glosseta, it contains more than 99% of all the Lepidoptera. 3 of the 4 suborders, they contain only around 250 known species. These are some of the common superfamilies of Dithyria: Hesperoidea, which includes keepers; Papilionoidea, which includes true butterflies; Geometridae, has mainly inchworms and loopers from family Geometridae; Bombycidae, mainly giant silk moths from family Saturnidae and sphinx moths from family Sphingidae; Noctidae, mainly cutworms and armyworms from family Noctidae; tiger moths from family Arctidae; and tussock and gypsy moths from family Lymantridae. Coming to the general morphology of insects, we can see that the body is divided into 3 distinct parts: head, thorax and abdomen. Okay, head consists of compound eyes, mouth parts and antennae. Okay, thorax consists of 2 pairs of membranous wings with few cross veins and 3 pairs of legs. Okay, 3 pairs of legs. Coming to the exoskeleton part, thorax is divided into prothorax, mesothorax and metathorax. Okay, and each segment consists of hardened plates or sclerites. Okay, dorsal is notum. Dorsal plates are called notal plates, and the ventral ones are called sternum, and lateral ones are called pleural. Okay, similarly, the 4 principal regions of abdomen, exoskeleton of abdomen, it includes same tergum, which is dorsal. Here it is notum, this is tergum. Okay, so tergum, ventral is sternal and lateral are pleural. Okay, and the reproductive parts, they include epiproct, paraproct, cercus and ovipositor. Okay, so this is the general morphology of insect. Coming to the structure of Lepidoptera, they also have same structure, like the body is divided into head, thorax and abdomen, as well as they will have eyes and antennae on the head. Thorax has pair of wings and 3 pairs of legs and abdomen. Okay, now this is, you can see, this is a butterfly and this one is moth. Now we will see the major difference between butterflies and moths. Okay, so you can see that usually you will get moths in night time and butterflies in the daytime, right? So butterflies are diurnal and most of the moths are nocturnal. Of course, whatever differences I am saying, there will be some exceptions, okay, to the rule. Second point is, butterflies are usually brightly colored and moths are dull in color, right? Third is, the antennae of butterfly is club shaped and that of moth is pectinate. Okay, we will see that later in the later slides. The front and the hind wings of the butterflies are held together in the flight by lobe processes at the base of the hind wing, which grips the underside of the front wings. Most moths have stiff bristles called frenulum at the base of the hind wing that hooks into the underside of the front wing. Okay, so it means moths have frenulum, butterflies have other parts and not frenulum. Next is, the scales on the wings of the moths have large scales, but that of butterflies, they have very fine scales. Main feature which you can see in butterfly is, butterfly folds wings vertically over their back, while moths hold wings horizontally when at rest. Next point of difference is, forelegs of the moths are fully developed and those of butterflies are reduced, missing terminal segments. Okay, and another difference which you can note is, thorax of moth is stout and that of butterfly will be thin, this way. Okay, and there will be some differentiation between the thorax and abdomen, which will not be seen in the moth. Okay, so these are some of the differences which you can observe in butterfly and moth. Coming to the difference between the male and female, you can see that abdomen consists of reproductive organs on the last segment. Okay, this is the last segment and it contains the reproductive organs. Males are generally recognized by two small protruding plate-like claspers. Okay, there will be, see, you can see here somewhat pointed thing. So they are protruding plate-like claspers located laterally at the tip of the abdomen, but female abdomen is larger and more pointed at the tips. Okay, so here you can see this is female one. Okay, the body, wings and appendages are clothed with broad scales, as I discussed it during the differences which I told, right? Scales are morphologically flattened and highly modified microtrachea. The colors of the scale pigments represent the butterfly coloration. Scale colors are derived from pigments and physical structure. The latter affects light transmission and diffraction due to particular arrangement of scales and the ridges and thin layered walls of scales. Coloration of a captured butterfly may have deteriorated due to loss of the scales. Okay, so if you see that scales are you are not able to see the scales properly, it means that they might have deteriorated. Okay, the different types of variations are seen in morphology of the butterflies, that is sexual, seasonal, individual and geographical. Okay, so morphologically also sometimes butterflies might have difference. Okay, on the basis of the sex, on the basis of season, okay, or individual and geographically. Now, coming to the characters which are used in identifying the lepidopterans. Mainly, these are some of the characters which are usually used for identifying the lepidopterans. Wings, antennae, ocelli, mouthparts, legs, size and color, and the anatomy of the reproductive organ. Okay, mostly with size and color itself sometimes we are able to identify. Mostly those who are not entomologists or layman, any common man will able to identify the butterfly with the size and color itself. Okay, not too much detail, but the common butterflies will be identified. Okay, for detailed identification you need to see all these other characters also. The shape and venation of the wings. Okay, whether the wings are homoneurous type, means the venation of the forewings and hind wings are alike, or heteroneurous type, whether the forewings and hind wings are different. Okay, whether the wings are aculeate, means more or less covered with specialized bristles called microsetae, or non-aculeate. The type of wing coupling, whether jugate or frenate. Okay, the structure of larva and position of primary setae, whether the pupa is axillate or octate. Here in our presentation I will be discussing mostly about adult characters. Will not be focusing on the larval and pupal characters, but just simple basic things I have mentioned here, whether the structure of larva and position of the primary setae, and whether the pupa is axillate or octate. Okay, here we have seen in the difference, I was saying difference between moths and butterflies, I was mentioning about the frenulum and all. So those wing coupling here we will be seeing for the characters for identifying the lepidopterans. These are the regions of the wings. Here you can see this is costa, this is forewing. First we will see this is forewing, this is hindwing. Okay, both have same parts. So what are they? So this is costa, this is apex, this is termen, tornus, dorsum, this is dorsum, this is base, and this is cell. Okay, and these are the regions. 8 is submarginal area, 9th is post-discal area, 10th is discal area, and 11th is basal area. Okay, similar parts are there in the hind wing also. First is costa, this is apex, this is termen, here this is tornus, this is dorsum, this is base, this is cell, this area is submarginal, this area is post-discal, this is discal, and this is basal. Okay. Seeing the general insect wing venation, we can see that the wing venation is not readily visible because of the covering of scales. Okay, when you have to put some drops of alcohol or ether allowed to fall onto the wings, and it will show up the veins for the few seconds. Okay, the variation of the wing venation are usually used for identification, and with regard to venation, wherever specialization is evident, it is due to atrophy or coalescence of the veins and not by addition. Okay, so whenever there will be difference, it will not be because of the addition of the veins, but might have the two veins might have coalesced. Okay, or there will be some atrophy in the veins. The general venation of insect wing is given here. Okay, so this is H is humeral. Okay, this is humeral, this is costa, the vein here is called costal vein, this area is subcosta, the vein is called subcostal vein, this is radius, it has one radial vein will be having branches, 5 branches mostly, so R1, R2, R3, R4 and R5. Okay, then media, the veins in this region are called medial veins, 3 medial veins, 3 branches of the medial veins you can see here, then cubitus, and then anal. Okay, these are anal veins, and these are interconnection between medial and cubital. This is radial and medial. Okay, so these are the interconnections. Coming to the region of the veins, wings, the part where the wing is connected with the thorax, that part is called humerus, and the angle is called humeral angle. Okay, this is costa or anterior margin, this is apical angle, this is anal angle. Okay, this is apical margin, this is anal or basal or inner margin. Okay, and this is discal cell. Okay, so most of the discal cells will be seen in this region. Then some will have jugal lobe, okay, and some will have anal lobe. An expansion of the area where there are the 3 anal veins. Now, first also we have mentioned about the types of venation. Here we will be seeing in the diagram. Homoneurous type. Homo means same. So front and hind venation is similar, and branches of R are equal in numbers in hind wing and forewing. Okay, similar, it means not all, but you can see the subcosta and radial as well as medial veins, they will be same. Okay, and like usually radial veins will be same in number. Okay, in both hind wing and forewing. Type of venation is usually seen in the species of Zeugloptera and Dicnonifer suborders. Second type of wing venation is heteroneurous type. Hetero means different. Okay, so hind wing venation is reduced. Okay, there will be less number of the veins as compared to the forewings, and RS is branched. Here you can see RS, this one, only one is there, it is branched, and this are 1, 2, 3, 4, 5. Okay, and this will only be 1. That's it. Okay, so these are the differences, major differences. You can see here A3 is also there, it's not there. This 3 median veins will be same. Okay, but major differences are radial veins. Okay, Monotricia and Ditrecia species, they will have such type of venation. Coming to the wing coupling apparatus. Okay, you might have seen always a butterfly will fly in, the wings will be in coordination. Okay, it like when the forewing will go up, the hind wing also will go up. Okay, from both the sides. Okay, similarly when the forewing will come down, hind wing also will come down. Okay, and it will create a flap. You will never see it like in coordinate manner. Okay, so that is due to wing coupling apparatus, which will connect both hind wing and forewing. Okay, to give the coordinated flight, right? So there are 4 general mechanisms whereby the wing of each side are operating together. These mechanisms, the parts are called a fibula, a jugum, a frenulum, and an expanded humeral angle of the hind wing. Okay, these are called wing coupling apparatus, which attains the high degree of specialization among various lepidopterans. Right. So first we will see fibula. Small, more or less triangular lobe present at the base of the front wing on the posterior side. This you can see here. Usually they are present in Zeuglopterans and Dicnonifer. Okay. Second type is jugum. It is usually present in monotricians, right? They are small finger-like globe. Here you can see this, present at the base of the front wing. Okay, and it overlaps the base of the anterior edge of the hind wing. Okay, so if this is the front wing, its base, okay, and it will overlap the hind wing below. Here will be the hind wing, and it will overlap the anterior edge, right? Like this is the anterior edge. So anterior edge of the hind wing will overlap this. Okay. Usually it is present in species of Monotricia. Coming to frenulum. They are the large bristles or the group of bristles arising from the humeral angle of the hind wing. Okay, humeral angle, here you can see this is humeral angle and it will come from here. So this one. Fitting under the group of scales near the costal margin on the lower surface of the front wing. So this is the costal margin, okay, and this is the lower surface of the front wing, this one. Okay, so this is the bristles arising from the humeral angle of the hind wing and fitting under the group of the scales near the costal margin of the lower surface of the front wing. Okay, most Monotricia and Ditrecia has, except few, has this type of frenulum. Okay. Next part is antennae. Butterflies, they have slender and knobbed type of antennae, and moths will have filiform, stachys and plumose, a plumose type of antennae. Butterflies, clubbed, this is pectinate, or filiform, you can say. Coming to the next feature, that is ocelli. Ocelli are simple eyes. Most of them have a pair of ocelli located on the upper surface of the head close to the compound eye. Most of the insects, they have compound eyes. Each compound eye will have cell, and number of cells it will form compound eye. A cell, they are called ommatidias. Compound eyes has more than 60,000 ommatidias. And these are the ocelli. Usually ocelli are simple eyes, right? And lepidopterans, they have only one pair of ocelli. And this third one, central one, is not there. Some families don't have ocelli at all, like Hyblidae, Lymantridae, Viponomutidae and Geometridae. These ones, they have usually ocelli, and this is Geometridae, they don't have ocelli. Coming to the mouth parts. The principal mouth parts are sucking tube or proboscis formed from the maxillae, which is held curled up in the spiral under the head when not in use. Here you can see. This proboscis is derived from the portions of the maxillae. It uncoils by hydrostatic pressure and acts as a siphon tube for sipping liquid nutrients such as nectar from flowers and other substrates. Now coming to the next character, that is legs. It includes the form of tibial spurs and tarsal claws. So first we will see the parts of the legs. Usual insect leg will have coxa, that is the base of the leg, the part where it will be attached with the body of the insect. Then trochanter, femur, tibia, tarsus, pretarsus, aerolium, this is tarsal claw, these are tarsomeres. And typical butterfly, lepidopteran leg, it will have epiphysis here in the front leg. So front leg will have epiphysis. So the presence or absence of spines on the legs and structure of the epiphysis is also regarded for identifying of the identification of a lepidopteran. So then the another thing was this epiphysis, they are usually movable pad or spur like structure on the inner surface of the front tibia used in cleaning the antennae. So these are usually present in the front legs. And you can see that front legs are very much reduced in Nymphalidae, Danidae and Satyridae. So these are the families where front legs will not be of the same size as other legs, other two pairs of the legs. So they will be very much reduced. Now coming to the unique morphological characters of Lepidoptera. So after seeing the characters individually, we can sum up. Summing is like wings with a dense scale covering on the veins and membrane, right? M veins of the wings with 3 branches instead of 4 or more, except in the only 2 known species of Agathophagidae. Then tibia of the 4 legs with only 1 or no apical spine, right? So if you would have seen the part of tibia in the foreleg, they will have one or no spine. Then loss of central ocellus, we have seen that. Okay, so simple eye but pair present. Okay, that central one is absent. Pair is it is in pair. Instead of 3, 2 will be there. First tergite very slightly sclerotized. Okay, so when there is sclerotization, that is hardening of the cuticle, it will become rigid. So to give the flexibility to the abdomen, the first tergite, that is the first exoskeleton on the dorsal side, it will be very, very soft. Okay, it will be very slightly sclerotized. It will not be very much hard. Okay, loss of cerci on the abdomen. Cerci will not be present in the lepidopterans usually. Then tibia on the front legs with epiphysis articulated on the inner surface. Okay, the last character which we have seen in the characters of legs. Now, if the butterflies are to be collected for now, usually we don't collect, but usually earlier times we were collecting them. They were appearing usually in largest number when there has been an abundance of larval food plant, like in spring from February to April, and during post monsoon tide from August to November. They live for the day. Normal lifespan is between 15 days to 3 weeks for small ones, and between month and 6 weeks for the bigger ones. Now these are the characteristics of suborders, like Glossetta. Okay, the one which is having maximum number of species. I will start with that. So they have vestigial and non-functional mandibles, an elongated galea forming a proboscis, also called hostilum. Why this change is there in them? If you see them, the food habit of a butterfly is type of sucking food habit. So they don't bite anything. So the mandibles, it has reduced, it has become vestigial. Okay, it is non-functional because it is not being used to cut or bite anything. Okay, so that mouth part, it has been converted to proboscis. Okay, or you can say hostilum, which is usually used to suck the sap from the flowers. Okay, nectar from the flowers, right? So this one change is there. Then larvae with the spinneret. Okay, an organ used to create silk thread, located on the top of the prelabial or hypopharyngeal lobe. Okay, and other 3 suborders, they have, the adults still have mandibles, functional in some group, atrophied in others, and ancestral. These are the ancestral that has been replaced by the proboscis in all other lepidopterans. Okay, so this is ancestral character, you can say. It is present in other 3 suborders, and advanced is in Glossetta. Okay, the presence of proboscis is advanced character for Glossetta. Then characteristics of 2 main clades. You can see Ditrecia have 2 separate sexual openings in the female, one for mating, okay, that is gonopore, and the other for egg laying or ovipore, and both have internal connection. Okay, second abdominal sternite with large apodems, muscles in the proboscis in short bands instead of long fibers. Okay, so these are the 3 main characters which differentiate Ditrecia from Monotrecia. Okay, Ditrecia lepidopterans have developed tympanal organ that enables them to hear the echolocation signals of the bats in order to avoid them. Okay, the more primitive families are collectively called the Monotrecia and have only a single sex opening. Okay. Coming to the major key for identification, this way, I'll just tell you the basic of how a key is made. I'm not going to the detail, okay, because it is too lengthy. If you all want to see it, you can see in the books of identification manuals of borer or INS. Okay, so these are some of the identifying characters which I am just mentioning in a very basic way. So usually identification will be done. First, characters which will be used will be wings, whether they are present and developed. If wings are present and well developed, then further front and hind wings are similar in venation or not, that will be seen. So if it is similar in venation, and usually also M shape, okay, and RS in the hind wing, 3 or 4 branched. Front and hind wings usually united by jugum or fibula and coiled proboscis. Okay, so these are some of the characters. Further, it will go to some other family. So these are the identifying key features which are used in the manual. Till this is till families. Okay, for the species, there will be further more identifying differences. Okay, so these are the different identifying characters which you can see in the key. Okay, so whether wings are absent or vestigial. If they are absent, then moth developing in and usually never leaving a sac or case constructed and carried about by larva. Okay, and another character is moth not developing in the sac or case constructed by larva. So further it will go to some other family. Okay, and this will be identified in some other family. This is the families of butterflies and moths can be known by observing their external. Now, due to decreasing numbers of lepidopteran species, the collection is not preferable. So it is always advisable to have a photograph of fresh specimen in their natural habitat. Okay. Ventinae, legs, wings, venation, they can be sketched. Okay, and by taking photographs you can do that. So these were the photographs which were taken by my reverend teacher, late Professor John Manchraker, okay, which I am using in my presentation. And some photographs I have taken from Wikipedia and other sites which I have mentioned in the okay. So these are the references from where I have taken this material to make my presentation. Okay. See you in our next video. Bye till then.